The Brahminy blind snake, a creature so unassuming it could be mistaken for an earthworm, is a fascinating subject of study. Its slender, shiny body often leads people to overlook it. But this tiny serpent is far more than meets the eye. Recently, it has been making headlines in the scientific community for reasons that go beyond its appearance. Researchers are delving into its biology to uncover secrets that could have wide-ranging implications. Don't let its diminutive size fool you. This tiny serpent harbors a genetic secret that has captivated researchers. Its scales, though small, hide a world of genetic information that is both unique and enlightening. Found in gardens and flower pots across the globe, the flowerpot snake, as it's commonly known, reproduces without mating, a phenomenon called Parthenogenesis. This means that females can produce offspring without the need for a male, a rare trait in the animal kingdom. What makes this snake even more intriguing is its unique genetic makeup. Instead of the usual 2 sets of chromosomes, it possesses 3. This triploid nature is a genetic anomaly that scientists are eager to understand. The Brahminy blind snake's unusual genetics offer a unique window into the complexities of evolution. By studying its genetic structure, scientists hope to gain insights into how genetic diversity and evolutionary processes work. This tiny creature challenges our traditional understanding of reproduction and genetic inheritance. Its ability to reproduce without a mate and its triploid chromosome structure are phenomena that defy conventional biological rules. By studying its peculiar adaptations, scientists hope to unlock new secrets about the mechanisms of evolution and the diversity of life on Earth. These findings could lead to breakthroughs in understanding how species adapt and survive in changing environments. The implications of this research extend far beyond the realm of snakes, potentially shedding light on genetic disorders and reproductive challenges in various species, including humans. Understanding the genetic makeup of the Brahminy blind snake could pave the way for new treatments and therapies for genetic conditions. As researchers continue to explore the genetic mysteries of this small but mighty snake, they are not only expanding our knowledge of the natural world, but also opening doors to new scientific possibilities. The Brahminy blind snake, with its unassuming presence, may hold the key to unlocking some of the most profound questions in biology and genetics. The Brahminy blind snake's possession of 3 sets of chromosomes, a condition known as triploidy, is typically associated with developmental abnormalities and infertility in most animals. In humans, for instance, triploidy is often lethal. Yet the flowerpot snake thrives, reproducing successfully through parthenogenesis. This unexpected resilience raises intriguing questions about the mechanisms that allow this snake to navigate the complexities of triploidy. How does it overcome the challenges of having 3 sets of chromosomes during development? The answers lie in the intricate dance of genetics, where the flowerpot snake has evolved unique mechanisms to maintain its genetic stability and reproductive success. Parthenogenesis, the ability to reproduce without mating, is a remarkable adaptation that allows the flowerpot snake to colonize new territories with remarkable efficiency. This unique reproductive strategy is not only fascinating but also incredibly advantageous in certain ecological contexts. By bypassing the need for a mate, the flowerpot snake can rapidly expand its population in new areas, ensuring its survival and dominance. A single female can establish a new population, eliminating the need to find a mate. This means that even in isolated or sparsely populated environments, the flowerpot snake can thrive. The ability to reproduce independently allows these snakes to take full advantage of any suitable habitat they encounter, spreading their presence far and wide. This ability makes the flowerpot snake a highly successful invasive species, often hitching rides in potted plants and quickly establishing themselves in new environments. Their presence in greenhouses and nurseries can lead to unintended consequences, as they outcompete native species and disrupt local ecosystems. The flowerpot snake's adaptability and resilience make it a formidable invader. While parthenogenesis offers clear advantages for dispersal and colonization, it also presents a genetic paradox. The lack of genetic variation can be a double-edged sword. On one hand, it ensures that successful traits are passed down without dilution. On the other hand, it means that the entire population is genetically identical, which can be risky. Asexual reproduction, by its very nature, limits genetic diversity. This limitation can make the population more susceptible to diseases and environmental changes. If a disease or environmental shift affects one individual, it could potentially impact the entire population due to their genetic uniformity. Without the mixing of genes from two parents, offspring are essentially clones of the mother, making them potentially vulnerable to environmental changes and diseases. This vulnerability highlights the delicate balance between the benefits and drawbacks of parthenogenesis. While it allows for rapid colonization and population growth, it also poses significant risks that could threaten the long-term survival of the species. Researchers at the University of Texas at Arlington and other institutions have been delving into the genetic secrets of the flowerpot snake, a fascinating species that has intrigued scientists for years. Their goal is to understand how this snake overcomes the significant challenges posed by reduced genetic diversity, a common issue in species that reproduce asexually. Through meticulous research, they have uncovered a remarkable adaptation. The flowerpot snake possesses highly efficient DNA repair mechanisms that are crucial for its survival. These mechanisms work tirelessly to correct errors that arise during DNA replication, a process that is prone to mistakes. By ensuring the genetic integrity of its offspring, these repair systems play a vital role in maintaining the health and viability of the species. However, efficient DNA repair alone doesn't fully explain the flowerpot snake's evolutionary success and resilience. Further research has uncovered another layer of complexity in the snake's genetic makeup. Despite reproducing asexually, which typically limits genetic variation, the flowerpot snake manages to maintain a surprising level of genetic diversity within its populations. This discovery suggests that the flowerpot snake may have evolved novel mechanisms to generate genetic variation, even in the absence of traditional sexual reproduction. These findings not only shed light on the unique biology of the flowerpot snake, but also provide valuable insights into the broader mechanisms of evolution and genetic adaptation in asexual species. Section 5. Triploidy's Human Connection Insights from Down Syndrome Research. The flowerpot snake's ability to thrive with 3 sets of chromosomes has piqued the interest of medical researchers studying human genetic disorders. Triploidy in humans, while rare, is a serious condition. Most cases result in miscarriage, and those that survive to birth typically have severe developmental abnormalities. Down syndrome, while not a complete triploidy, is caused by an extra copy of chromosome 21. Studying how the flowerpot snake manages its extra set of chromosomes could provide valuable insights into the mechanisms of triploidy and potentially lead to new avenues for research into human genetic disorders like Down syndrome. Section 6. The flowerpot snake's genetic legacy, unraveling evolutionary mysteries. The seemingly insignificant flowerpot snake has emerged as a powerful model organism for studying the complexities of genetics and evolution. Its ability to thrive with triploidy and reproduce through parthenogenesis challenges our understanding of these processes. By unraveling the genetic secrets of this tiny serpent, we gain a deeper appreciation for the diversity of life on Earth and the remarkable adaptations that allow species to survive and thrive in a wide range of environments. The flowerpot snake's story reminds us that even the smallest and most unassuming creatures can hold the keys to unlocking some of biology's biggest mysteries. Section 7. Beyond the flowerpot, Parthenogenesis in a wider context. The flowerpot snake is not alone in its ability to reproduce through parthenogenesis. This phenomenon has been observed in a variety of species, from insects to reptiles to fish. Each species that has evolved this ability offers a unique opportunity to study the evolutionary pressures that drive the development of such an unusual reproductive strategy. Understanding the commonalities and differences in the genetic mechanisms underlying parthenogenesis in various species can shed light on the evolutionary history and ecological factors that have shaped this intriguing reproductive mode. Section 8. A Future of Discovery. The uncharted territory of snake genetics. The flowerpot snake's unique genetics have opened a new chapter in our understanding of snake evolution. As we delve deeper into the genetic makeup of these fascinating creatures, we can expect to uncover even more surprises. Future research will likely focus on identifying the specific genes and molecular pathways involved in the flowerpot snake's DNA repair mechanisms, and its ability to maintain genetic diversity despite asexual reproduction. These discoveries could have far-reaching implications for our understanding of genetics, evolution, and the interconnectedness of life on Earth.